Now that I've messed with your eyes long enough, let's go ahead and start. And we'll finish up with influenza this morning, more than you ever wanted to know about flu. So we left off talking about the fact that influenza A is actually a virus that's predominantly found among birds. But there are influenza viruses for a number of species, including dogs and horses. But the unfortunate thing is that because of this segmented nature of the genome of influenza virus, you can have arising what are referred to as reassortments. So this is especially prominent coming out of pigs, because pigs, remember, have two different kinds of sialic acid receptors, one being very similar to human sialic acid receptors and the other being similar to avian receptors. So there's the possibility, in fact the probability, that pigs can be simultaneously infected with a human influenza virus and a virus from some other species. And despite the fact that this picture makes it look as if the second virus is transmitted rectally, it is not. These are both respiratory infections of the pig. And so what can happen is that in one individual cell of the respiratory tract of that pig, you can have the RNA segments from two different influenza viruses, one from a bird and one from a human. And so they go through the entire replication process that we went through on Wednesday. And remember that the other key point is that as the virus buds from this cell, it has no specific means to assure that any virus gets even 8 segments, let alone which 8 segments. So when you have the simultaneous infection with two different viruses, what can come out of that infected cell is a virus that has exactly the same 8 segments as the human virus, or one that comes out with 8 segments of the avian virus. But probably those are the rare events coming out of that simultaneously infected cell. Almost certainly some different arrangement of RNA segments occurs, and most of those are going to be selected against by natural selection. So most of the viruses that have like 3 bird fragments and the rest human fragments might not actually be able to replicate even in the pig. But every once in a while, something happens where particularly one segment, and it's usually either the segment that encodes the hemagglutinin or the RNA fragment that encodes the neuraminidase. Those can get swapped, so that now what can come out of that pig may be a virus that has a segment that's never been seen before in the human population. And so humans won't have any immunity to that particular virus, or at least not the protein made by that virus. So this can lead to what's known as an antigenic shift, where suddenly, one segment of a virus now circulating in the human population has never circulated in the human population before. So one of the things that allows us to survive influenza, even though we can be infected over and over again, is the fact that we have some degree of immunity against it. And that level of immunity might not be sufficient to stop the infection from beginning, but what it does do is that it limits the severity of the influenza. So those of you who have, like many of us, complained that you got an influenza vaccine and then turned around and got the flu, you think that that's a complete failure of the vaccine, and in fact it's not. More than likely the disease that you got was less severe simply because you had some level of immunity. But now, if you have zero immunity against the hemagglutinin molecule from an avian virus that now can infect you, the disease can be much more severe and may actually infect a different part of your respiratory tract, especially the lower portions of the lungs. So these kind of antigenic shift events are how pandemic strains occur. So we really worry about the co-infection of pigs as a mixing bowl. Because horses, as well as birds and pigs and dogs, ferrets, seals, there's a lot of influenza viruses that can infect and can lead to this kind of reassortment. Now that's distinct from a process known as antigenic drift. I mentioned in the early part of the lectures on influenza that this viral replicase, RNA-dependent RNA polymerase, is a sloppy enzyme. It doesn't do a particularly good job because it can't proofread. No RNA polymerase that I know of has proofreading activity. And so, on average, every virus that is coming out of an infected cell, on average, should have at least one nucleotide substitution somewhere in one of those 8 genome fragments. And with the number of viruses that come out of there, virtually every possible mutation is probably occurring. What that does is, over time, is that allows a slow accumulation of mutations as the virus spreads throughout the world. And so this is another reason why we have to be boosted periodically for the same strains of influenza that are circulating in the human population. Because as they circulate, even though they might not acquire a new segment of RNA, they do acquire mutations which change the amino acids in the proteins. And so our immune system may no longer be quite as effective at stopping or preventing a severe form of the disease. So this antigenic drift is due to the fact that viral RNA dependent RNA polymerase is a pretty sloppy enzyme. Now, just to kind of show you something that will kind of drive home the point of antigenic shift, this is the genesis, the evolutionary genesis that has been placed or that has been put together for the 2009 pandemic swine flu. So what we know now is that the 2009 pandemic strain actually is a reassortment of 4 different viruses. So, but prior to the advent of this virus, there was a classical swine virus predominantly being spread among pigs that was a hemagglutinin-1 neuraminidase 1. Somehow, probably infecting another pig simultaneously, a North American strain of H1N1 flu infected the pig at the same time, as did a human influenza with an H3 or hemagglutinin-3 neuraminidase. So this kind of reassortment, it may not have happened all at one time. You know, you could have initially had a reassortment that occurred in a pig between the classic swine strain and the human strain, and then that virus may later have recombined with the bird strain circulating in North America. But in any case, what came out eventually was a different form. And so you can see these are color-coded. So this first, the intermediate virus that eventually leads to the pandemic strain has got 2 RNA segments from birds. It's got 4 strains from pigs and 2 strands from humans. Now that actually infects another pig at the same time that a different strain, a Eurasian swine-like flu, infects. And so you get another reassortment. This is the one that actually ends up being the pandemic that spread throughout the world. And so if you look at this, you'll see that fragment 4 is from swine flu. Fragment 6, the neuraminidase, comes from the avian-like swine flu. Fragment 2 is the only fragment left behind of the original human virus. And it's perhaps very fortunate that it was fragment 2 that was the one human fragment left. I mentioned that prior to this swine flu pandemic in 2009, all of the major pandemics that caused huge amounts of death in the previous century all had PB1 frame 2 peptides. Those were always coming from birds. So for some reason that people don't quite understand, the polymerase gene, this PB2 gene, is very important for virulence. And because this particular virus happened to retain the human form of PB1, it doesn't encode that small peptide, which induced apoptosis by targeting the mitochondria. So we were just actually that close to another potential pandemic that may have killed, it could have killed as many as 20 or 50 million, kind of like the 1918 flu. We don't really know. But we're always that close to the next major pandemic. And the problem is, from a public health point of view, is that people have been hearing this so many times that they, it's like the boy who cries wolf. People thought that, they heard about this terrible swine flu pandemic that was occurring in 2009. But then, since there wasn't like the major death toll that people were predicting, now people have become kind of dulled to the calls of public health officials. And hopefully none of us will ever actually have to experience the 1918 flu-like pandemic. But it's really fascinating to see how people have been able to piece together that influenza epidemic. As it turns out, and I think I mentioned this in earlier slides, there are 16 known hemagglutinin types, and only 3 of them have ever been shown to replicate in humans and spread from human to human. So, and we named them, obviously, the top 3, H1, H2, and H3. And only 2 neuraminidase molecules will function in a virus that passes from human to human, neuraminidase 1 and neuraminidase 2. So if you look at these major pandemics, and these pandemics have frequently replaced the previously circulating form of influenza. So for instance, this 1918 pandemic flu pretty much disappeared, at least the specific type, and was replaced by H2N2, which was the Asian flu in the early 1950s. And then the H3N2 strain tended to replace that largely. So we still have multiple strains circulating in the human population even now. The Russian flu was an interesting flu in 1977. This actually is believed to have been a weaponized form that accidentally was released from a Soviet weapons infectious disease factory somewhere near the Chinese border, but that's never actually been proven. But conspiracy theorists like to claim that this was the Russian flu was genetically identical to the H1N1 that caused the 1918 pandemic. But luckily enough, people were still somewhat immune, that the disease wasn't quite as severe. Now, there is currently circulating in birds, and this is the cause of worry for a lot of public health microbiologists, these high pathogenicity, or HPAI, strains of avian influenza. That's what HPAI stands for, high pathogenicity avian influenza. This is the H5N1 strain, which is actually occurring today as an epizootic and is actually a panzootic. So this is, I bring these words up for semantic reasons, and it also tells you a little bit about how we as humans place ourselves above all other animals. The term epidemic that we use is actually a word that's restricted only to humans. So epidemic means a disease circulating in humans. If a disease is circulating in any other creature, it's not called an epidemic. Then it's called an epizootic. And if that disease now affects multiple species throughout the world, it's referred to as a panzootic, not a pandemic. Pandemic is a term that we reserve only for diseases spreading among humans. All other animals are categorized as epizootics and panzootics. Now, this isn't something that you need to know, but the public health professionals survey the world every year looking for and trying to predict what viruses are going to cause the outbreaks of flu that year, and they try to manufacture particular vaccines. So for this particular year, the vaccine, the trivalent vaccine that most of you are going to be boosted with, is going to have 2 influenza A viruses. So when you see this kind of terminology, you can actually understand a little bit about it. The vaccine that has 3 components has 2 influenza A's, one isolated in California in 2009, and it's an H1N1 virus. The other one was isolated in 2014 in Hong Kong, and it's an H3N2. So these are the viruses that are predicted to cause epidemic, potentially pandemic forms of influenza. And these vaccines almost always have an influenza B, which we haven't talked about very much. Influenza B does not infect animals. It infects only humans. And so it doesn't get the reassortment that influenza A does, so it doesn't get as much press. However, it still causes, a very severe, oftentimes fatal disease. So the particular B virus that's currently being formulated into vaccines was isolated in Australia in 2008. And there are vaccine formulations that will have a fourth virus in it, which will be an influenza B virus isolated in Thailand in 2013. So when you see this, you can actually read what, you actually know what the information is. And so this was just from the CDC website. Okay, so that's it for influenza. That's more than you probably ever wanted to know about influenza. Now we're going to move on to the last virus that we're going to talk about in this class. It's another class of RNA viruses. These are the retroviruses. So retroviruses actually fall into three families. And for the most part, we're only going to talk about one. But there are families, there's a family called spumavirinae, which is a retrovirus that I don't think anyone knows anything about. So we won't even mention it more than that. There's another one that we'll talk about in another slide very briefly called oncorna viruses. These are retroviruses that cause cancer. Very serious diseases, obviously, but at least among humans, retroviruses that cause cancer are pretty rare. So we won't spend much time talking about them. For the most part, when we talk about retroviruses, because what we're interested in is human disease for the most part, right? We're talking about lentiviruses. So L-E-N-T-I viruses are slow viruses, lenti meaning slow. So these are the typical viruses like HIV-1, HIV-2. They're called slow or lentiviruses because the disease takes years to develop after the infection. But all three of these families of retroviruses all have a very similar structure in terms of the fact that they are, like influenza, an enveloped virus. So in this particular picture, you can see here a lipid bilayer that was acquired from the previous host cell that was infected. And that lipid bilayer is now studded with glycoproteins. So in this particular case, if we were talking about HIV, we would be talking about GP41, this transmembrane protein, and GP120. But all retroviruses typically have one single glycoprotein, viral envelope protein. It's usually processed into two proteins from one larger protein, and we'll talk about that. But then as you move internally into these viruses, what you'll see is that inside in a nucleocapsid made up of proteins encoded by the virus, you'll find two identical copies of a plus-stranded single-strand RNA genome. So this genetic material is very similar to the genetic material in polioviruses. It is a single-stranded RNA genome. It is of the positive sense, meaning that it is in the same sense as messenger RNA. And in fact, it could be translated into protein immediately upon infection of the host cell. But it's not. And we'll come to exactly why that is. This RNA segment actually has a 5' methylguanosine cap. So as I said, it could be translated immediately upon infection, but it's not. This virus is a little bit unusual in that it's diploid. There are always two identical copies of the entire RNA genome of retrovirus contained within this capsid. What the second copy is for is still a little bit controversial. We'll talk about one of the theories as to why there's a second copy of this RNA genome, and it has to do with its replication cycle. But I think still to this day, no one knows precisely why that occurs. But the other things that it's important to remember that the retrovirus has with it in this infectious form of the virus is that it has several enzymes that are present preformed within that virus. Probably the one that gets the most press that you wouldn't be surprised when I told you is reverse transcriptase. This is why we call these retroviruses. So David Baltimore discovered the fact that these viruses, despite the fact that they have an RNA genome, they have an enzymatic activity that can turn that RNA genome into double-stranded DNA. So that was very much against central dogma, right? So central dogma had always claimed that information biologically flowed from DNA to RNA to protein. Here was the first example of something that defied central dogma. Here was an enzyme that could reverse transcribe. It could take RNA and turn it into DNA. It had never been seen before. That enzyme needs to come preformed in that cell, or excuse me, in that virion. So this is different than the poliovirus. Remember, the poliovirus has this same conformation of an RNA genome. It's plus-stranded. It does not need to bring viral replicase with it because it is translated from its genome. In other words, that genome in the poliovirus, when it gets dumped into a susceptible cell, will be translated into a protein that will be processed and made into viral replicase to make more copies of that genome. That doesn't happen in retroviruses, and you'll see why. But so this virus has to bring reverse transcriptase with it. It also brings, very importantly, with it another enzyme made by the virus in the previously infected cell called integrase. That name tells you something that you really already know about retroviruses. They have the ability to integrate their double-stranded DNA genomes, once reverse transcriptase has made them. They can integrate that DNA into the host cell genome. And they also bring with them a proteolytic enzyme, a protease, which is involved in maturation. It's a protein that's actually responsible for cleaving, for instance, these outer membrane proteins, these glycoproteins. So these are actually two separate proteins, yet they were initially synthesized as one larger protein. And this protease has been responsible for cleaving those proteins. So there are several enzymes that it's important that you remember come with the virus at the time of infection. So all retroviruses, by definition, have to have at least 3 different genes. And some of the simplest retroviruses actually only have 3 genes. So in a typical retrovirus, in a very simple one, the only 3 protein encoding genes are referred to as GAG, PAL, and ONV. GAG stands for group antigens. These are genes that encode the structural proteins that make up the capsid. The PAL, you could probably guess what that is. That's the polymerase. That's the gene for reverse transcriptase, and actually an integrase as well is part of this polymerase gene. And then ONV stands for envelope proteins. So those glycoproteins that stud the lipid bilayer of that envelope are encoded by the ONV gene. I mentioned that some viruses in the retroviral family can cause cancer. And so in those viruses, there's always a 4th gene. Probably prior to the advent of HIV and AIDS, most studies of retroviruses were involved in veterinary science. So Rous sarcoma virus was one of the first viruses known that was capable of causing cancer, and it causes cancer in birds. So what's known about this virus is that it has a 4th gene called SARC for sarcoma, because this virus causes a type of tumor in birds called sarcoma. The SARC gene here is actually a degenerate or highly evolved form of a normal host cell gene that somehow in the course of the evolution of this oncovirus has now been incorporated into the retroviral genome, and it's been modified. So we won't talk much about viral oncogenesis, but an oncogene like SARC, whose genesis is normally or has come from a host cell, now has simply lost the ability to be regulated. So SARC is going to cause kind of an uncontrolled cellular replication of an infected cell. Kind of like the cellular analog of SARC would cause cellular replication, but in normal situation in the cell, that would be regulated properly so that the cell would not replicate in an uncontrolled manner. This gene has lost the ability to be regulated in that way. And so These cause cancer. Now, since people, and there are examples of oncoviruses in the retroviral family that do cause human cancers. Some leukemia viruses are retroviruses. Fortunately, they are rare. They occur and they're tragic for the people that get them, but because the diseases are so rare, there's not a large amount of research going on in the oncoviruses that cause human cancers. Most of the retroviral research in humans and most of what we know about retroviruses comes after the advent of HIV and AIDS. So let's just first, we're going to go through in a very detailed, perhaps too detailed, description of the replication of retroviruses and HIV specifically. But let's just first go through a very simplified overview of the replication process of a retrovirus. So here's a viral particle with its RNA genome. It should be represented as two of them here at the top, but there's really only one. Reverse transcriptase, that enzyme that's present within the viral particle, will, after infecting the cell, turn that RNA genome into a double-stranded DNA genome. And it's going to, at the same time, generate what are known as long terminal repeats. We'll talk a lot about long terminal repeats before we're done with retroviruses. That form, that now double-stranded copy of the viral genome will now be inserted randomly within the host DNA. So the reverse transcriptase has generated the long terminal repeats. These, you can tell from the name, these are longer than the original repeats. What I probably should have mentioned, and I forgot to, in this particular slide, was some other characteristics of these retroviral genomes that don't encode proteins. So in this retrovirus, you'll see on the left-hand end and the right-hand end, terminal direct repeats, these R sequences. Just inside of that, you'll see a sequence called U5, which stands for unique 5' sequence. So this is a sequence that doesn't code for protein and is found only on the 5' end of the genome, as opposed to the unique 3' sequence, which is just internal to the right-hand repeat sequence. So this repeat sequence at the RNA level, once retrovirus gets a hold of it and completes it, changes the nature of that repeat sequence and it lengthens it. So that now at the DNA level, that sequence is now referred to as the long terminal repeat, or the LTR. So those long terminal repeats are generated, and those long terminal repeats are critical for the function of retroviruses because they help to do two things. First of all, they help the viral DNA to incorporate into the host genome. And as you'll see later, the long terminal repeats now, and only now, once they've been turned from repeats to long terminal repeats, now contain very strong promoters that's going to allow the host RNA polymerase to transcribe this viral DNA genome into viral messenger RNA. So that's what's shown here. You do not get transcription of the virus messenger RNA until it's been turned into double-stranded DNA and inserted into the host. Once it's inserted into the host now will bind its RNA polymerase to the long terminal repeats to generate very typical messenger RNA. The host has no way to know that this messenger RNA is actually toxic to the cell. It looks just like a host messenger RNA because, in fact, it is a host messenger RNA. This viral RNA genome, this provirus now, as far as the cell is concerned, is just another set of genes in that genome. This messenger RNA that's made will now encode all of the proteins to make the nucleocapsid, as well as the proteins that are going to stud the lipid bilayer, which is going to make the envelope. So in a process very similar to the influenza process, where we talked about hemagglutinin and neuraminidase and the M2 protein being trafficked via the endoplasmic reticulum and the Golgi apparatus to rafts on the membrane of the cell, that's the same thing being represented here in this little decurved section. These little black spots are meant to represent the viral glycoproteins now embedded in the host membrane, and now the virus will bud off from that and be released. Kind of similar to the lambda situation in terms of terminology, the virus is now referred to as a provirus as opposed to prophage in lambda. And it incorporates randomly. And then once transcription and translation have reached a sufficient level and the timing is proper, then the encapsulation will begin and the viruses are released by budding from the cell. So the cell is not lysed directly, but the viruses just bud off. So this is, again, similar to influenza. Now, so that's just a general quick overview of the process. Now let's go through it starting from the beginning and talk about some of the important steps in. Now we're going to switch and talk about HIV instead of talking about retroviruses in general. Largely what I want to talk about now is HIV because that's what we're really interested in here, right? Is that here is the HIV virion and a potentially targeted cell. And some of this is not going to be new to you, but these viral glycoproteins that exist in the lipid bilayer envelope now have the ability to bind to not one, but they must bind to two receptors on a host cell. So what dictates tropism in HIV, and by tropism I mean what is the tissue type that can be infected, that tropism is determined by the array of membrane proteins and receptors that a cell can produce. This GP41, or glycoprotein 41 of HIV, binds to can bind to three different proteins. It always binds to this protein CD4, and it has to have a co-receptor besides that. So it either has as its co-receptor a protein called CCR5, which is found predominantly on macrophage and dendritic cells, so cells of our nonspecific immune system. These are cells that surveil our mucosal membranes for pathogens to try and stop infections before they begin. So macrophage have this CCR5 protein on the surface, and probably about 1/3 of T lymphocytes also have this. But the GP1 or the GP41 or GP120 rather also has the ability to bind to a different co-receptor if that's present as well, called CXCR4. That's found predominantly on T cells. Most all T lymphocytes or thymus-derived lymphocytes possess this form of the co-receptor. So the natural ligand, so these proteins did not evolve in order to allow HIV to bind and infect those cells. Those proteins actually have natural agonists or natural ligands. So in the case of CCR5, the macrophage and dendritic cell glycoprotein, it normally binds chemokines. So chemokines are chemicals that are released by other cells, especially if they've been infected, to now attract macrophage or neutrophils or other cells to sites of infection. So infected cells will release things like RANTES or MIP1 alpha. These will induce white blood cell or leukocyte migrations towards them. That's the normal agonist for CCR5. Now, there's actually a new drug. I don't know how new it is at this point, but there's a drug on the market now that actually helps to limit infections. So this is a drug called Maraviroc. This is an antagonist to CCR5. So in other words, it can bind to CCR5, but not have the same effect as the agonist. So in other words, if you treated this cell with a chemical like RANTES, that would induce a lot of physiologic changes within this cell because that's the agonist for CCR5. Maraviroc is a chemical that is an antagonist because it can bind to the CCR5 and it can block HIV from binding to it. So this is the kind of drug that would be given to somebody who is HIV positive, but this would potentially limit the infection of new cells. So as viruses are released within that person, the virus wouldn't be able to infect another cell. This is also potentially a drug that would be given prophylactically. So a healthcare worker who may have been exposed accidentally in a healthcare setting to blood or bodily fluids from an HIV positive person, as part of the prophylaxis, this drug would make sense because it would potentially block the virus from infecting that person's macrophage cells. So it's potentially a very important drug. Now, this CCR5, this co-receptor, this chemokine receptor, has a really interesting history. It was actually discovered in the 1990s by a really clever postdoc at Rockefeller University named Rong Liu. He was working in the lab at the time at Rockefeller in Nathaniel Landau's lab, who's still a big name in HIV. But Rong Liu noticed in doing some studies that the lab was doing that there was a cohort of men in New York City that were taking part in the study that, despite the fact that they were reporting having unprotected sex with people who were known to be HIV positive, these men were never, not only were they never coming down with AIDS, they were never becoming infected with the HIV, with HIV. They seemed to be completely resistant to it. And for the longest time, nobody would listen to this postdoc saying that there was something different about these people. What he did find out eventually, because he did the experiments himself, and the paper was published in Cell in 1996, it was a major, earth-shattering discovery, because what he found was that there was a group of men, this same group of men, that had homozygous mutation where they had deleted, or they didn't delete them, but they had two alleles of the gene for CCR5 that had a 32 nucleotide deletion within the gene. So they had two alleles of what's referred to as CCR5 delta 32. If you subtract 32 nucleotides from a gene, that's not in multiples of 3, right? So that results in a frame shift. So what happens is that a person who has two alleles of CCR5 delta 32 does not produce that protein. So what happens is that this slide is not it's good and it's not good. It shows that in a person who's homozygous for the delta 32 allele, there is no CCR5. They are still producing the CD4, that other co-receptor. That should be shown here. But without that important CCR5 co-receptor, people who were homozygous for this mutation were resistant to HIV infection. At least by normal routes. In the laboratory, you could actually infect their macrophage, completely artificially with different forms of HIV, and we'll talk about the different tropic forms. But what people realized was that other groups, when they began to look at this study a little, or these groups of people a little more closely, that there were other people who were in fact being infected, but they weren't developing the disease until much later in the course of the infection. And they found that these people were heterozygous. So they were producing only half as much of the CCR5. So the progression of the disease in people who were heterozygous for this was much slower than people who were homozygous for the wild type form that produced normal amounts of this chemokine receptor. So it's a really fascinating story because what we now know, or it's still controversial, is what has selected for this deletion in the CCR5. Because if you look at human populations worldwide, you'll see as much as 20% of the human population from Northern Europe, and I mean like Scandinavian Northern Europe, about 20% of that population will be at least heterozygous for delta 32, and about 1% to 2% will be homozygous for it. Whereas you virtually never see this allele in Asia or Africa. It's arisen entirely in Northern European. So for the longest time, people thought, because there was a connection, that some of these people may have survived the bubonic plague. For the longest time, people thought, and there was very good evidence for this, and I actually fell into this camp myself, that there was such strong evidence that said that people with the delta 32 deletions were more likely to survive infection with Yersinia pestis, the bacterium that caused bubonic plague, that was the positive selection. And so that this mutation actually benefited those people. What we know now, or what we think we know now, and I think I'm much more convinced of this, that it wasn't bubonic plague that provided the selective pressure. It was more likely smallpox. As it turns out, smallpox virus, we're not going to talk about pox virus, it's that DNA virus, actually uses CCR5 as one of its co-receptors as well. Smallpox is a much better selective agent from an evolutionary point of view because smallpox kills people before they reproduce. For the most part, if people have reached the age, have survived back in the 1300s and 1400s, if people survive long enough to reproduce, they probably survived smallpox as children. So they were immune. Those people were potentially carrying the deletions of this Delta 32. Because if the people who were dying from this, who didn't have that deletion, were being killed before they reproduced. So smallpox is a much better selective agent. So now I think what most people tend to accept, even though you can't really do the experiments, right? Because first of all, smallpox is extinct now. Second of all, it would be highly unethical to start trying to infect people with smallpox. But what most people think is that was because smallpox virus uses the CCR5 as its receptor, that's probably what allowed for one population throughout the world to deal with smallpox. The thing that we still don't understand is why it is that the same mutation didn't arise in Africa, didn't arise in Asia. Because there are other groups of people who are spontaneously immune to HIV. So there are other genetic mutations that have arisen in different parts of the world, independently of CCR5, that also provide protection, probably against smallpox, but also to some degree against HIV as well. So it's just a really fascinating evolutionary biology and public health story. So I love that story. So in terms of how this infection actually proceeds, in this particular case, we're talking about likely a macrophage, because here is the HIV virion with its lipid bilayer. It has two glycoproteins, GP41 and GP120, anchored within the lipid bilayer of that enveloped virus. GP120 and GP41 were, at one point, one protein. These were proteolytically cleaved into two proteins. And again, this is very similar to the hemagglutinin story in influenza. Very, very similar. Because if there is not a proteolytic cleavage between GP120 and GP41 from the precursor protein, which is called GP160, the math almost works out. If you didn't have that proteolytic cleavage, this infectious process that we're about to describe could not occur. Very similar to the cleavage that occurs in hemagglutinin. Without that cleavage, you couldn't have membrane fusion. So here, the GP120, the surface protein, actually has the ability to bind, in this case, to CCR5 and simultaneously to CD4. When all three of those proteins come together, and it does require all three of them to come together at one time, there is a major conformational change in the viral protein, such that the SU protein, the surface protein, or GP120, falls off. The conformation of the remaining portion of the protein, the GP41, changes so much that actually a portion of the protein of GP41 is now transported from the viral envelope into the host cell membrane. Again, that conformation change brings those two lipid bilayers so close together that they actually contact each other and will fuse. This is exactly the same situation that we just described in the last lecture for hemagglutinin in influenza. But now we're talking about the fusion between HIV virion and the membrane of this dendritic cell or macrophage cell. So now that can spill into the cytoplasm, that infectious material. Now, that's how macrophage cells get infected. And so most new infections, most sexually acquired infections of HIV, the initial cell that is targeted are macrophage cells. And this makes sense because the virus is going to be introduced at mucosal sites, and one of the primary cells that protects our mucosal surfaces from incoming viruses, parasites, and bacteria are macrophage. So That occurs early on in infection. Later in infection, in that infected individual, about 60% of people who are infected with HIV within several years, because they'll be healthy for a while, will begin to produce a different form of the same virus. So the virus actually evolves within a human. It partly has to do with the human's immune system. It partly perhaps has to do with the virus destroying enough of the macrophage dendritic cells such that it doesn't have enough targets anymore. But at some frequency, a virus comes to be inside an infected person, which now no longer quite so effectively binds CCR5, but instead acquires the ability to bind to CXCR4. X4, as it's referred to for short, is a protein that's present on all T cells. So whereas CCR5 was only on maybe 20% or 30% of T cells, so most T cells could not be infected. Most of those important immune cells could not be infected by what's referred to as an R5 tropic virus. The initial virus that caused the infection targeted a macrophage. Later on in the infection, now the evolution of these R4 tropic viruses now gives the virus a whole new set of cells that it can infect, the T cells. This is why people don't typically see the results of AIDS until several years after the infection, because the initial virus that's being produced in that cell or in that person is probably not very efficient at infecting lymphocytes. It infects only a small fraction of them. Later on in the infection, once this new form evolves, all of those T cells can be infected. And so that's probably when you begin to see the most profound effects of HIV/AIDS, because now a lot of the T lymphocytes are being infected and lost. Now, we can't really do a very good job at treating or using an antagonist against CXCR4. So I mentioned that Miraviroc that pharmaceutical companies have come up with to block CCR5 interactions with HIV. That doesn't seem to have too much negative effect on the human physiology. However, this R4 binds to a chemokine called stromal-derived factor 1. And that actually plays a major role in stem cell homeostasis. So drugs that actually potentially serve as antagonists to stop the binding to T cells actually have some pretty profound negative effects. So this is not something that we can interfere with too much chemotherapeutically. This just kind of shows them side by side. So on the right are the macrophage or R5 tropic strains of HIV. So this would be early on in the infection. It binds to CD4 as well as the CCR5 to initiate infection. So 90% of all HIV-1 infections, new HIV-1 infections, are the result of this R5 tropic strain. It's not that people can't be infected with the strains that arise later, these X4 tropic strains. But these viruses, are not as easily bind to macrophage cells. These are viruses that are now binding to T cells. So sexual encounters don't tend to introduce viruses now that are capable of binding macrophage. Instead, they're capable of binding T cells. So it's a much less likely occurrence. Plus the fact that probably after several years before the development of these X4 tropic strains, people are perhaps so ill, and they know that they're so ill, they're perhaps either using barrier methods to protect their loved ones, or perhaps are just abstaining completely. So 90% of new infections are due to these M-tropic or R5 viruses. The X4 tropic ones come up much later. So those infected macrophage cells during the initial infection have the ability to travel to regional lymph nodes, and they can actually facilitate the infection of T cells, those cells that have CCR5, which is about as many as 30% of T cells. But those cells may regenerate. The T cell tropic ones, or the X4 tropic ones, arise in about 60% of people who are infected within about 5 years. The percentage rises further on than that. But at 5 years, about 60% of people now are producing two kinds of viruses, or maybe they have perhaps switched from CCR5 tropic to CXCR4 tropic. And actually, it gets even more complicated because there are viral lineages that have been isolated that actually can bind to both co-receptors simultaneously. So this is not a switch from on to off. This is a slow evolutionary process. And so we'll pick up with this when we come back on Monday.